Good morning. I'm Christian Hinrichs. I'm an assistant clinical investigator at the National Cancer Institute. I'm an immunologist and medical oncologist. This is federally funded research that was conducted at the NIH Clinical Center. The title of the study is HPV-targeted tumor-infiltrating lymphocytes for metastatic cervical cancer. While it's hoped that vaccines and screening will reduce the future incidence of cervical cancer, at present time in the United States it causes the deaths of more than 4,000 women each year. Chemotherapy for advanced stage cervical cancer is not curative and it rarely provides durable palliation, so better treatments are desperately needed. Cervical cancers harbor attractive therapeutic targets for immunotherapy in the HPV E6 and E7 oncoproteins, but clinical trials of immunotherapy in this disease have been disappointing up to this time. Adoptive T-cell therapy is an emerging and promising immunotherapy platform that can cause complete and durable tumor responses in metastatic melanoma and certain B-cell malignancies. But its study in epithelial cancers really has been very limited, and it has not been studied in cervical cancer. So this is the treatment schema for how this type of therapy is performed. We call it HPV-targeted tumor-infiltrating T-cell therapy, or HPV-TIL. From a patient with metastatic cervical cancer, we excise a tumor. T-cells are cultured from the tumor fragments. The cells that grow out in each of those cultures are then tested for reactivity against the HPV E6 and E7 antigens. The reactive cultures are preferentially selected and they're expanded for administration to the patients. The cells are administered in a single cell infusion. Prior to cell infusion, the patients are treated with a lymphocyte depleting chemotherapy regimen consisting of cyclophosphamide and fludarabine. After the cell infusion, they're treated with aldesleukin, which is dose to tolerance. This slide summarizes the patients treated thus far. We've treated 9 patients. The age, as is typical of cervical cancer, is generally quite young. The histologies are primarily squamous cell carcinoma and adenocarcinoma, entirely squamous cell carcinoma and adenocarcinoma. And the HPV type has been HPV-16 and HPV-18. Now in our cohort of patients, we've seen more patients with adenocarcinoma and the HPV-18 type than what's generally seen. The prior systemic therapies are variable, but I want to point out that some of these patients are very heavily pretreated, including 2 patients who had objective clinical responses to the treatment, patients 2 and 3. The cell dose has ranged from 33 to 152 billion cells, and the clinical responses have occurred in 3 of the 9 patients. 1 patient had a partial response of 3 months duration, but 2 patients had complete responses, one that was ongoing at 18 months and the other at 11 months at the time that I made the slide. Now both those patients followed up last week, and those numbers can now be extended to 22 months and 15 months. This slide shows the imaging studies from the 2 patients who had complete responses. Patient 3 was a 36-year-old woman with a metastatic squamous cell carcinoma of the uterine cervix. She had been previously treated with 3 different combination cytotoxic chemotherapy regimens. She had multiple tumor sites, as you can see indicated by the yellow arrows, and she had a complete response to the treatment with no evidence of disease at 18 months, and her scans from last week at 22 months look the same. Patient 6 was a 36-year-old woman with a HPV-18 positive adenocarcinoma. She had a very aggressive primary tumor that was refractory to chemoradiation, and then at the time of surgery for her primary tumor that did not respond, was found to have spread of her disease in the pelvis and at distant sites. At the time that she received our cell therapy, she had multiple sites of disease indicated here by the yellow arrows. I'll also point out the red arrowhead, which points to a ureteral stent that's holding open her ureter due to compression from the neighboring tumor. She had complete regression of all sites of disease, and her ureteral stent has been removed, and she's now getting along normally. Here you see 11 months after treatment. They're the same at 15 months. So in conclusion, this study shows that durable, complete tumor regression can occur following a single infusion of HPV-targeted tumor-infiltrating T cells. So this study provides proof of principle that an immunotherapy can induce regression of cervical cancer and that adoptive T cell therapy can mediate regression of an epithelial cancer. We believe these results warrant continued study of HPV TIL for patients with metastatic cervical cancer. Thank you for your attention.